So there's some other inheritance patterns that occur besides the complete dominance that we've been looking at. So we're going to learn about incomplete dominance, codominance, multiple alleles, epistasis, and also polygenic inheritance. So we've been talking about complete dominance, where the dominant allele will mask the recessive allele and it'll determine the phenotype. So if you have an individual whose phenotype is purple, it could be either homozygous dominant or heterozygous. And then if its phenotype is a white flower, its genotype is going to be homozygous recessive. But complete dominance is not always the case, and we can also have incomplete dominance and codominance. So let's take a look at those. All right, so with incomplete dominance, when you have the heterozygote, it's going to have an intermediate phenotype between the homozygous dominant and homozygous recessive phenotypes. So it actually looks like blending is occurring, but actually that is not happening. So let's take a look at some examples here. All right, so here's an example of incomplete dominance. So we've got a red flowered plant and a white flowered plant. So when we cross them, if incomplete dominance is occurring, we get offspring that are pink. And so it looks like the red and the white alleles blended together to make the pink offspring. So it looks like blending, but it actually is not. Because if you went and crossed two pink individuals, here what their offspring would be. So the red and the white alleles would reappear. So those red and white alleles have not blended together. So they're still there. So it's not blending. So this is what we call incomplete dominance. And you can see how we're writing. The alleles is a little bit different. So sometimes with incomplete dominance, we can write them as our allele key. We would have our flower color gene, and we would say the color gene, the red allele, would be designated by that, and then the white allele would give us that color. And notice that they're both capital letters, because neither one is actually dominant over the other one, because the heterozygote, if you have both a red and a white allele together, would actually give you a pink individual. Okay, whereas the red individual would have to have two red alleles, and then the white individual would have to have two white alleles. Okay, another way that you could do this is you could have your allele key, and if you wanted to, you could say, well, the capital letter could be red, and then the lowercase letter could be white, and then you would just have to remember that because neither one is actually dominant over the other, if you have two big C's together, that would be a red individual. And if you have the heterozygote, that would be pink. And then if you have, whoops, and then if you have two lowercase c's, they would be white. So you could write it that way if you wanted, but just knowing that, you know, the big C is actually really not dominant over the lowercase c. okay, because this is incomplete dominance. So the big C is not going to mask the little c. It actually looks like the red and the white blend together to give pink, but they're actually not blending. So the red and the white alleles are still there, but what's happening is to get the pink color, this color allele is producing an enzyme to make the red pigment. And you have to have 2 copies of the red allele to make enough of the enzyme to get the red color. And then if you only have 1 copy of this red allele, you're not making enough of the enzyme to get a fully red color, and you end up with a pink color. And then if you've got a white individual, they're not able to make the enzyme, and so they don't make the red pigment, and that's why they're actually white. So that's actually what's happening in incomplete dominance. Okay, and then we have codominance. So in codominance, the heterozygote is actually going to express the phenotype of both alleles. So a good example of this is in human blood types. So in human blood types, there's actually 3 alleles. So there's an I with an A, that's 1 allele. There's I with B, that's a 2nd allele, and then there's little I. So that's your 3rd allele. So any one person, because you're diploid, you can only have 2 of these alleles. You will never have all 3, because if you had all 3, that would mean you're triploid, and you'd have 3 copies of a chromosome, and you probably wouldn't be alive. So you're only diploid, and you can only have 2 of these alleles. Okay, but there are 3 in the population. Okay, and 2 of these alleles are codominant to each other. So the IA and the IB alleles are codominant to each other. So that just means that neither one is dominant to the other. And so both of them are going to be expressed. Okay, so that means on your red blood cell on the surface, these alleles are going to code for a protein. So the IA allele will code for the A protein, and then the B allele will code for the B protein on the surface of these red blood cells. And this is what's going to cause this person to be type AB blood, because they will express both the A and the B protein on the surface of their blood. So that would be codominance. And then the little I allele, the reason we have a lowercase is because it's going to be recessive to the A and the B alleles, okay, which are going to be dominant to the little I. Okay, so if we look at the blood group types, if you have type A blood, these could be your possible genotypes. You could have 2 of the A alleles or an A and a little I, and you would only produce the A protein on the surface of your red blood cells. If you have type B blood, these are the possible genotypes. You could have both B alleles or a B and a little I, and you would produce the B protein on the surface of your red blood cells. And then here's this codominance example. If you have the genotype AB, so you'd have type AB blood, you would produce both the A and the B protein on the surface of your red blood cells. So you would express both phenotypes. And then if you have type O blood, your genotype would be the 2 little I's. So you're going to be homozygous recessive here. And when you have type O blood, you do not produce any proteins on the surface of your red blood cells. Okay, and then here's just another example of codominance, and then we'll compare that to incomplete dominance. So remember, incomplete dominance, neither allele is completely dominant over the other, and it looks like blending. So if you have a red and a white flower and you cross those, you get the pink flower. And then codominance, you're going to express both of the phenotypes. So you would see both the red and the white alleles. And so your flower, you could see both the red and the white alleles being expressed. So it wouldn't be a pink flower, but you'd have red and white patches on them. And so that would be codominance. So incomplete dominance looks like blending. You would get an intermediate phenotype between the red and the white, which would be pink. And then codominance, you would express both the red and the white, and so you would get patches of both of those colors. Okay, and then here's just another slide, just talking about the difference between complete dominance, incomplete dominance, and codominance, and their examples. All right, and then another different inheritance pattern we have is multiple alleles. So a lot of our genes can have more than 2 alleles in a population. So blood type would be a great example of multiple alleles. So with blood type, we have 3 alleles in our population. But remember, any one person will only have 2 of the alleles because we're diploid. So we can only have 2 of these alleles. You will never have all 3 of them in one individual person. But in a population of people, you know, if you checked everyone's blood type, you probably would find all 3 alleles in the population. But any one person will only have 2 of these alleles. Okay, and then here's just a nice summary with examples here of complete dominance, incomplete dominance, codominance, and then we've also added the multiple alleles as well. Okay, and now we're going to take a look at when we have 2 or more genes and how that affects inheritance. So we've got epistasis and polygenic inheritance. So let's look at epistasis first. So the word epistasis is kind of a funny word. So if we just look at the word epistasis, so epi means upon or above, and stasis means to stand. So it literally means that one gene is going to stand on top of the other, or one gene is going to affect the expression of another gene. So a really good example of this is in Labrador retrievers. So we've got 2 genes here that will affect the coat color of the labs. Okay, so in our labs we have black labs, we have chocolate labs, and we have yellow labs. Okay, so one gene is going to determine whether the lab will be black or chocolate lab. So black will be dominant over the chocolate color. And then the other gene, this E gene, will tell you whether you can deposit the color or not. So the dominant gene says, yes, you can put down the color, whether you're supposed to be black or chocolate. And then the recessive gene says, no, you can't put that coat color down. So if you're homozygous recessive for that E gene, that means you won't put the black or chocolate color down at all, and the coat color will be yellow. Okay, so let's take a look at epistasis. All right, so here are the possible genotypes for if you were a black lab or a yellow lab or a chocolate lab. And notice for the yellow lab, this E gene is homozygous recessive, and it says, I don't care if you're supposed to be black or chocolate, you're not putting that color down and you're going to be yellow. Okay. All right, so here we've got two parents. Both of them are black labs, and notice that we're doing a dihybrid cross. So we are looking at two genes. We've got a coat color gene where they're going to be black or chocolate, and here we've got two that are black. And then this E gene is telling you whether you can put the coat color down or not, and this says yes, you can put the coat color down. Okay, and they're hybrids, they're heterozygous for both of these genes. Okay, and remember in dihybrid crosses, we would expect to get a 9:3 to 3:1 phenotype ratio because of that law of independent assortment. Okay, so when we look at our possible gametes that we can make, we could have big B going with big E, so that would be one gamete. We could also have big B going with little e, that's another gamete. We could have, we'll go over here just for more room, we could have little b going with big E. There's another gamete. And then we could also have little b going with little e. Okay, so this is this law of independent assortment. So these chromosomes can line up different ways in metaphase 1, and we can have these two genes, these alleles, ending up with different partners between the B's and the E's. Okay. All right, so when we cross these are all the possible offsprings we can get. And notice that with epistasis, we don't get our 9:3 to 3:1 ratio. We get a 9:3 to 4 ratio. And the reason that happens is because of that E gene. Okay, so we've got one gene affecting the expression of the other. So remember, if they're homozygous recessive, it says, hey, you can't put that coat color down. Okay, even though you were meant to be chocolate, you can't put that chocolate color down and you're going to be yellow. And that's why you get this 4 here instead of a 3:1. So 9:3, and then this kind of collapses into a 4 instead of a 3:1. So that's epistasis. You just have one gene affecting the expression of another. Okay, and then our last one is polygenic inheritance. So again, if we look at the name, poly meaning many, and gene is talking about genes. So we just have more than one gene affecting a single trait. Okay, so for example, if we look at skin color or height. Okay, so for example with skin color, there's actually 60 different genes that affect your one single trait, your skin color. Okay, and we end up with what we call these more or less traits. Okay, so you've got lots of different skin colors. You can be more dark, less dark. Okay, and so you kind of get this bell curve with the different skin colors. Okay, and the same thing happens with height. All right, so we're going to simplify skin color and just simplify it down to 3 genes instead of 60. Okay, so with our skin color, we're just going to look at these 3 genes. And we've got our A, B, and C genes. And the capital letters mean you're going to have darker skin, and the lowercase letter means you're going to have lighter skin. And so with 3 genes, we've got this giant Punnett square with 64 possible offspring. Okay, and these parents are heterozygous for all 3 of these genes, so they would have a medium skin tone. And you can see that with their offspring, most of them are going to be medium tone as well, so similar in skin tone to the parents. So you have the greatest probability of having a similar skin tone to the parents, but it is possible, a 1 in 64 chance of getting a really light skin tone and a 1 in 64 chance of getting a really dark skin tone with those two parents. Okay, so this is from that Punnett square, the probabilities, the number of offspring we would get with those two parents. And so you can see most of the offspring would be the same skin tone as the parents, but it is possible to get a very light or a very dark offspring. And then here's just an example here. We've got two fraternal twins. They're not identical, but two separate eggs, two separate sperm that fertilize these two eggs. And you can see the difference in skin color with these two sisters. So same parents, but you can see with this bell curve, it is possible to have different skin tones from those two parents. Okay, and then this is just a nice summary looking at the difference between epistasis. So you've got one gene affecting the expression of another gene, and then polygenic inheritance, you've got several genes affecting one trait. So for example, like skin color or height.